AP 120, Chapter 4, topic is translation. So, the messenger RNA leaves and enters the cytoplasm in order to produce a certain protein. However, when we look at proteins, we see they have 20 different amino acids. But RNA, and DNA, only have 4 nucleotides. So how do you get from 4 nucleotides to 20 amino acids? Well, what was finally discovered by scientists is that you have to break up the RNA into 3 nucleotide chunks. And those 3 nucleotide chunks are called codons. And so each one of these 3 nucleotide sequences is coding for a specific codon or a stop signal. So it is the messenger RNA's codons, the sequence of these codons, that leads to the sequence of the amino acids. And this, of course, all occurs in the cytoplasm with the help of the lovely ribosome. So here's the messenger RNA with its sequence. Here is some of its codons. Here's the ribosome. Ribosome possesses rRNA. We, of course, have amino acids that we're going to need, and also tRNAs. So it is the tRNAs, the transfer RNAs, that bring the amino acid to the ribosome. And the way the system makes sure that the correct amino acid is brought is that the tRNA has a 3 nucleotide sequence called an anticodon that needs to complementary base pair with the codon. And that way, only the correct tRNA is able to base pair and bring in the correct amino acid. The thing is connected to the growing polypeptide. So again, the anticodon must complementary base pair with the codon. So UAC in the tRNA is base pairing with AUG in the messenger RNA. Sequence of codons determines protein sequence. All right, so it starts with the ribosome coming in and attaching to a messenger RNA, finds the beginning of the important sequence, and then tRNAs come in, and if the tRNA's anticodon matches with the codon, then it stays in and has brought the correct amino acid. The ribosome will then form a peptide bond between the new amino acid and the growing polypeptide chain. And then the ribosome will move down the messenger RNA to the next codon. So the ribosome is moving down along the messenger RNA in 3 nucleotide chunks in order to get to the next codon and the next necessary amino acid. This lovely figure in your textbook gives the whole story of protein synthesis, talking initially about transcription occurring in the nucleus, the messenger RNA leaving, and then translation occurring in the cytoplasm. I recommend you review that. All right, so we have now figured out which codons code for each of the amino acids. And we're now able to use this lovely chart to take messenger RNA sequence and turn it into amino acids. And you will need to know how to use this chart. It'll be a question where you're given the chart on the exam and also in one of the lab quizzes. So, starts off, you've got your codon. So here's the messenger RNA codon. We always want to use messenger RNA for this chart because that's how it's designed. So, first nucleotide in the codon is A. So we come over here to the first letter. Here is A. So now we know that the amino acid is found somewhere in this strip of options on the chart. The second codon in this case is U. Here is U for the second letter. Go down, we see that they get overlap here. So the amino acid must be within this section of the chart because of the overlap. And then the third letter, the G, here's G. Bring it all the way over and look at that. Here is what we have, AUG, the amino acid methionine, which is also the start sequence for translation. So again, first letter along here, second letter along here, third letter along here, always looking for where all three overlap to give us the new amino acid. All right, so here is mRNA sequence. What is the first thing that needs to be done? I hope you said break it up into codons. That is correct. So we break it up into 3 nucleotide chunks. Then we have to figure out what the amino acids are. So I recommend you pause the video and determine what the 3 amino acids are using this chart. All right, I hope you got serine, histidine, and glutamic acid. Just so you know, you don't need to learn the names of all the amino acids. It's be supplied on the chart. So serine UCG, first letter U, so it's in this strip, second letter C, so it's in this area, third letter G. Look at that, UCG gives you serine. Histidine, CAU, first letter C, so it's in this area, second letter A, so it's in this area now, third letter U, there's the U, so CAU, histidine, and so on. You will notice that some amino acids get coded for by multiple codons. That's perfectly fine. Some amino acids only have one, and there are some stop sequences. So when the ribosome reaches a stop sequence, it removes the messenger RNA from its internal workings and is done making that protein. All right, here's another sequence. This is DNA sequence. You know that because it has T's in it. What is the first thing you must do? Well, I hope you said do complementary base pairing to turn it into messenger RNA. So again, if you start with DNA sequence, you have to do complementary base pairing to make the messenger RNA sequence. Then, after that, what do? I hope you said break it up into the codons. That is correct. And then you take the sequences and you use this chart to give you the proper amino acids. I recommend you pause the video and do that. All right, I hope you got these amino acids. Methionine, again, AUG. Glutamic acid, so first letter C, here's C, second letter A, here's A, third letter A, here's A, CAA gives you glutamic acid, glutamine. And then isoleucine, first letter A in this area, second letter U, so it's over here, third letter U, AUU, isoleucine. All right, mutation. As I mentioned earlier, mutations happen sometimes just by accident. DNA sequence sometimes is permanently changed during DNA replication. And sometimes mutations occur because of exposure to mutagens that damage the DNA. The mutation is a permanent change in the DNA sequence. In normal body cells, it will get passed on whenever those cells divide. And then those cells can accumulate more mutations over time. If it's a sex cell, if it's a sperm or oocyte that is mutated, then it gets passed on to the children. The mutations can be bad, they can be silent, or they can be good. So, one kind of mutation is a point mutation. With a point mutation, a nucleotide in the DNA sequence does not have its complementary nucleotide brought in. There's a 0.1 single nucleic acid that is now gone. Point mutation. I'm sorry, I lied. Let me start over. Point mutation, you get a change in the DNA, so one single nucleotide is different, which means you now have a different nucleotide in the resulting messenger RNA. Again, point mutations, a single nucleotide is changed in the DNA, a single nucleotide in the DNA is different, and that leads to a different nucleotide in the messenger RNA. Break it up into codons. We see the sequence. Here's the one that's now different. Figure out what the amino acids are. Hope you pause to do that. And then here is the resulting amino acids. Now our original sequence gave us methionine, glutamine, and isoleucine. This time we get methionine, histidine, and isoleucine. So we have a different amino acid, one amino acid change in the protein. This could have a huge effect, could have no effect. It's hard to say. So that is a point mutation, single sequence change in the DNA leading to a single change in the messenger RNA, which leads to a change in the one amino acid. Here is another point mutation. So the C is going to be a G now. Break up the messenger RNA into the codons. Look at the resulting amino acids and look at that. Originally methionine, glutamine, isoleucine. Now we have methionine, glutamine, isoleucine. This is a silent mutation. Even though the sequence in the DNA and messenger RNA is different, it still leads to the same amino acids. So there was no change in the protein being produced. And again, this is a consequence of the fact that some amino acids have multiple codons, multiple sequences that can code for them. All right, here is a frame shift mutation. Frame shift mutation is when a nucleotide in the DNA is completely deleted, meaning that that nucleotide doesn't have its complementary base pair in the RNA, so a complete deletion, or a complete addition of a new nucleotide. So here's the deletion from our sequence that we've been using. Break the messenger RNA up into codons, pause so you can figure out the amino acids. And the correct amino acids are serine, lysine, phenylalanine. Now we look, and we're in our original sequence, methionine, glutamine, isoleucine. So look at that. All three amino acids are now different. So with a frame shift mutation, not only do you change the sequence of that codon, where the missing nucleotide would be normally, but it changes the sequence to all the rest of the messenger RNA. So all the rest amino acids are probably going to be different than what they were before. So frame shift mutations also change amino acids downstream from the mutation, and it's a huge deal. Now, you can also do this with insertions. You could have an additional nucleotide added to the DNA sequence, which means there's an additional nucleotide in the messenger RNA, which then causes the frame shift mutation. So point mutations, only one nucleotide is changed to a different nucleotide. Frame shift mutation, an entire nucleotide is removed from DNA, or an entire nucleotide is added to DNA. Which of these mutations will have a more dramatic effect on the protein? Hope you said frame shift mutation, because it disrupts all the codons at the mutation and downstream of that mutation, which affects all of the amino acids at the mutation and downstream of that mutation. All right, here is another lovely sequence. It is DNA sequence. You know this because you see those T's. So what is the messenger RNA sequence? I will pause to let you figure that out. Hopefully you got this. Now, what is the amino acid sequence using this lovely, lovely chart? Pause the video again so you can do that. Here it is. Woo! So if you were successful in doing this, then you know how to use this chart. And again, like I said, it's going to show up in the exam and also in a lab quiz. All right, that is it for this chapter.